when the cell just divides, the meiosis II starts. As you can see in the diagram, because now we know that after meiosis I, one cell is divided into two cells, each with a half set of chromosomes, each with a half number of chromosomes. In case of human beings, for example, if there were 46 chromosomes in 23 pairs, then each daughter cell after meiosis I now will have 23 chromosomes, one from each homologue. In case of an organism, for example, which have a 16 total chromosomes in 8 pairs, both daughter cells will now have 8 chromosomes each. That is one set, and all are recombined. So recombination is done. Now both of these cells will enter into the meiosis II. Meiosis II, so it means that meiosis II will occur in the cells, in the two cells which are already produced by the meiosis I. You can see the first cell is dividing and the second cell is also dividing. The meiosis II is just like the mitosis. In the meiosis II, first phase is prophase. We call it prophase II because this is the prophase of the meiosis II. In prophase II, the chromosomes, they appear, they become visible under the microscope because of the condensation, because they are condensed, they are thickened, and the centrioles start moving in the animal cells. Centrioles start moving towards the poles and they start making the spindle fibers. And again, at the end of the prophase, the nuclear membrane will disappear. At about end of the prophase II, the nuclear membrane disappears and the chromosomes and spindle fibers are there in place in the center. After the end of prophase II, metaphase II starts. In the metaphase II, the chromosomes now arrange themselves on the equatorial plate. Now, as we know, they are duplicated and now each sister chromatid is attached with the spindle fibers with the help of separate kinetochores, like that of mitosis. And they arrange themselves on the equatorial plate. As you can see, this is observable in both cells that they are arranged on the equatorial plate, and now each sister chromatid is separately attached with the spindle fiber. Then comes the anaphase II. In anaphase II, the chromosomes start separating from each other because the spindle fibers, they start pulling these chromosomes towards the poles, and now because each sister chromatid was separately attached to a spindle fiber by its own kinetochore, each sister chromatid moved towards the poles. So the result is an equal number of chromosomes distributed towards each pole. When the anaphase II ends, then comes the telophase II. In the telophase II, the chromosomes in equal numbers now reached at the poles, nuclear membrane starts forming, nuclear material start decondensing again and become invisible as that of the interphase. The spindle fibers, they are deformed, they are degraded, and each cell is divided into two, makes total of the 4. So in meiosis I, what happened? That one cell is divided into two, each with a half number of chromosomes, but both of these cells, which were the products of the meiosis I, were individually divided by another phase of meiosis called meiosis II, and the end products of these two cells is 4 cells, all with a half number of chromosomes, because reduction is already complete in the meiosis I. So this is how by one mother cell in the germline cells is divided into 4 cells, 4 daughter cells, either sperms or eggs, which have a half number of chromosomes in each. There is another difference in the meiosis between the males and the females. In males, one germline cell divides into 4 equal sized cells. It means that one germline cell makes 4 sperms with a haploid number of chromosomes, n number of chromosomes. But in females, there is a difference. As you can see in the diagram, when after the meiosis I, which is the reduction division, chromosome number become 1/2. But when the cell divides, it divides unequally. It means that chromosome number is half in both of the cells, but the cytoplasm and the cytoplasmic contents goes more towards one cell, and the other cell though have equal number of chromosomes but have fewer parts of cytoplasm and the others, which results in one cell which is large, which is called a secondary oocyte, and the other is called a polar body, which is very small. Next division, in the next division, the polar body again divides, but it divides, that is in the meiosis II, it divides into equal size cells, but the secondary oocyte, that is the other cell which is larger in size, is again divided by meiosis II, but this division is again an unequal division. The result is in a very large ovum and a second polar body. So it means that in a female, the cells, as you can see in the diagram, one cell, the primary oocyte, is divided into 4 cells, but 3 of these are called polar bodies, which are small in size and which are discarded, and the one which is larger in size is called the ovum, and this is retained for fertilization. So there is a difference between the meiosis in males and females in this regard.